Hello and welcome to the second recorded lecture for microbiology for the surgical technologist. In this chapter, we're going to talk about the science behind microbiology and so some learning objectives. We're going to talk about some key terms and we're going to distinguish between normal flora and pathogenic microbes. We're also going to compare the various classification systems of living organisms, discuss characteristics of prokaryotes, describe how viruses and prions become pathogenic, and discuss the systems of nomenclature and taxonomy to classify microbes. All right, so now we're going to talk about what is microbiology. In the last chapter, we talked a lot about who's contributed to it and how did we get from the abiogenesis theory of spontaneous generation to now we know that there's these little creatures we can't see with our eyeballs, and they're the ones that are responsible for causing disease, right? If a microbe causes a disease in a human, it's said to be pathogenic. And so microbiology is the study of these little creatures that for the most part we can't see. And so what does that include? Well, that includes bacteria, archaea, algae, fungi, protozoa, and viruses. And we want to stop these little guys in their tracks when we're in surgery. And we don't want to introduce any microorganisms into the field whatsoever because this could potentially cause our patient a surgical site infection. And so controlling these microbial populations is of great importance to us so that we can provide our patient with the best possible outcome. Microbial life was here way before we were here. In fact, scientists have been able to find evidence of microbial life dating back to more than 3 billion years ago. And so they're the ancestors of life on Earth. But, they're the reason that we have diseases. And these diseases, we refer to them as pathological conditions, but they're also responsible for helping us live a normal life, right? Bacteria and various microbes do a lot of good things for us. For example, we have something on our skin called normal flora. And in the surgical realms, we refer to this as resident flora, right? It's the bacteria that lives there all the time. And we have 10 bacteria for every cell of our body. There are 10 bacteria in us or on us at all different times, and they're helping us, right? So this resident flora or normal flora, it lives on us all the time and it helps us. And then the flip side of that is transient flora. Transient flora is that bacteria that we pick up from around our world. All right, so some bacteria that might help us, for example, live inside the digestive tract are called E. coli. And, they're really helpful, they're really awesome, but if they get out of their normal environment, their microbiome, then they can wreak havoc. E. coli, for example, if it ends up in the abdominal cavity, can cause a serious infection that could ultimately lead to death. Suffice it to say that scientific classifications have undergone a lot of revisions over the years. In 4th century BC, Aristotle attempted to classify living things, and he said that there were 2 kingdoms, plants and animals, end of story. And then a German biologist came along, and in 1894 he said, well, along with plants and animals, we also need to represent the smaller creatures that we can't see, and so he called that kingdom Protista. And so that included single-celled eukaryotes and then bacteria, which we refer to as prokaryotes. It underwent some more changes, and then there was a 5-kingdom classification system. And with that 5-kingdom classification system, we ended up with plants, animals, protists, monera, and fungi. And then Dr. Woese, he came along in the late 1900s, 1990, and he came up with this 3-domain system. And the 3 domains are bacteria, archaea, and eukarya. And, over time, scientists have tried to study the genomes and, link animals together by looking at their DNA and so on and so forth. And one interesting thing that Dr. Woese said is he said that, if we were to get rid of all multicellular life forms, the microbial life might experience a little bit of a change. But if the reverse were to happen, that life on this planet would cease to exist as we know it. And so really, it's these microbes that are literally supporting our entire ecosystem and allowing us to thrive. And here's just an image representing the 3-domain system that the text talks about that include, as I said, bacteria, archaea, and eukarya. And you can see within each of these kind of breaks down bacteria as cyanobacteria and chloroplasts, which chloroplasts are kind of plant-like, and gram-positive bacteria and gram-negative bacteria, and so on and so forth. All right, so now we're going to take some time to discuss these different domains and some organisms that we have mentioned already in our lecture. So first we're going to talk about eukaryotes. And when I think of eukaryotes, I think of us. They're multicellular organisms for the most part. There are some single-cell eukaryotes, but what define eukaryotes is that their cells have a nucleus, and inside the nucleus is DNA. Right? That's the biggest distinguishing characteristic of eukaryotes. Prokaryotes, on the other hand, encompass all bacteria. All bacteria have RNA that is just kind of floating in the cytoplasm, and it is not contained by a membrane-bound nucleus. And then we have archaea, and archaea is the newest form of microbial life to be studied, and they are the oldest known prokaryotes on the planet. They're single-celled, and they share a similar structure with bacteria, but they have some distinct genetic differences. They show an affinity or a high liking to extreme environments, like in the depths of the ocean where we have these volcanic vents and it's hundreds of degrees. They love to live there. They also love to live in glaciers and geothermal springs and salt marshes. So while eukaryotes, prokaryotes, and archaea are all living organisms, there are some non-living pathogens as well, and those are viruses and prions. Now, viruses we've talked a little bit about before. They either have DNA or RNA, and they are quite a bit smaller than viruses. They can be, or than bacteria, excuse me. Viruses can be up to 10,000 times smaller than some bacteria, and they're referred to as acellular. And what this means is they just consist of nucleic acid, and that nucleic acid is wrapped in a proteinaceous coat called a capsomere. And some of them also have an additional wrapping or additional envelope, and these are called retroviruses. And retroviruses utilize and spread only RNA genetic material and are responsible for HIV and those types of diseases, human diseases. So viruses are sometimes referred to as obligate intracellular parasites, and what that means is that they can't reproduce on their own, so they're going to have to find a host cell that they can invade. Remember, they're like the pirates of the microbial world, and so they're going to invade host cell, commandeer their machinery, and go about the business of replication. Okay, or reproduction. Prions are a proteinaceous material, and they are responsible for causing something called mad cow disease in cows, but in humans they cause something similar called Creutzfeldt-Jakob's disease, CJD, and this pretty much, CJD has the same symptoms as Alzheimer's disease. However, it can take years and years to manifest itself, so people may not even know that they have had it. There was a rash of it in Europe, and so if there have been individuals that are visiting there, they could have been exposed or at risk of developing CJD. What makes it really bad is that we don't have any way to kill it. And so if we operate on a patient that we know has CJD on their brain specifically, because it has an affinity for brain tissue, then we're going to need to discard all of the instrumentation that we have used. So we'll try to use as much disposable stuff as possible, you know, but we don't really have a way to kill them because most of the sterilization methods that we use denature the nucleus and the proteins inside the nucleus and allow it to, or render it unable to replicate itself. So with prions, they don't have that. They're just this protein thing. So killing it is very difficult. From the scientific classification, so those three domains that we talked about, bacteria, archaea, and eukarya, we also use a classification system called taxonomy. And taxonomy is used to establish relationships between like organisms, differentiate between two groups of organisms, or classify a previously unknown organism. And you can see the taxonomic hierarchy here on the left, starting with kingdom at the bottom is the widest, broadest category. And then we work up from there, phylum, class, order, family, genus, species. And this one here is specifically referring to the human species. So you can see that the human, it belongs to the kingdom, the animal kingdom. The phylum is Chordata, which means we have a backbone. The class is mammal. We have fur or hair and milk glands, so that classifies us as a mammal. The order that we belong to is primates. We have collarbones and grasping fingers with opposable thumbs, which is awesome. Family is hominids. So primates with relatively flat faces, three-dimensional vision. Thank you for the rods and cones. And then our genus is Homo, so hominids with upright posture and large brains, yes. And then lastly, our species would be Homo sapien, which is a member of the genus Homo with a classified as a high forehead and thin skull bones. Okay, so that name that you see at the top, Homo sapien, that is referred to as binomial nomenclature. And so it's a two-part naming system, and you need to write it in italics. So Homo sapien, for example, would be written in italics. And the first part is the genus, Homo, and then the second part is the species, Sapiens. All right, so there you have it. Scientists unanimously agree that one of the most challenging areas of microbiology is that of identifying and classifying viruses. They are non-living and they are so incredibly small. Remember, they can be up to 10,000 times smaller than a bacteria. That there's a special microscope, an electron microscope, that we have to use to be able to see them. And so some methods of classifying them include their morphology, which is their size, shape, and arrangement, as well as the type of nucleic acid they have. Remember, they can have DNA or RNA, how they replicate, the types of hosts that they invade, and the type of diseases they cause when they invade those living organisms. And so there's 3 general classifications that are based on the types of cells that they penetrate, and that includes zoophaginae, phytophaginae, and phaginae. And so there is this committee, interestingly, the International Committee on Taxonomy of Viruses, and there are 6 subcommittees, and these subcommittees are responsible for these viruses. And each committee is responsible for one type, whether it's fungal viruses, plant viruses, invertebrate viruses, prokaryotic viruses, and vertebrate viruses. And then there's one committee that just manages data and websites. So when we classify viruses, when I say we, I mean they, there is not a domain or a kingdom included because they are not alive, but as far as taxonomy, they do have an order, a family, a genus, and a species. And there's some various ins and outs to that. There's also something called a Baltimore classification system, and David Baltimore devised a system for classifying viruses, and he divided them up into 7 groups on the basis of their messenger RNA. And so there are some different classifications he used there, some characteristics to determine which 7 groups they fit into. All right, everyone, this concludes our lecture on Chapter 2, The Science of Microbiology. And I know with every lecture, you are going to fall more and more deeply in love with microbiology.